Good morning to all. So I am very glad to invite Dr. S. Triveni for delivering a guest lecture on plant genetics for modulation of crop microbiomes. Many of the students will not be knowing about her. She, as Dr. Sukumar said, she graduated from Andhra Pradesh and also postgraduate completed from Department of Microbiology, AC&R in Madurai, under the chairmanship of great microbiologist Dr. Anthony Roy, sir, and she is heading the department in Hyderabad. I had a great association with the department. Previously, Dr. Subhash Reddy was head of the department. From the days onwards when he was associate professor, I used to visit the department frequently for several occasions. And when I conducted her for this guest lecture, she readily accepted. So it is our visionary thought, our Honorable Vice Chancellor. She wants every faculty who is coming inside to TNIS to deliver a guest lecture for the benefit of the students. At least few students, why not few, all students can get benefited by this lecture. But due to various pressure on classes and other thing, it is very difficult. Usually we used to arrange on Friday. Every Friday we earmarked as endowment lecture and guest lecture. We made a free in the attendance also. But today is being Thursday, it is very difficult to manage some of the classes for some of the students. Anyhow, those who are here will get benefited by the lecture. I am also interested to know about some of the facts on this her lecture also. So I thank Dr. Triveni for accepting the offer and delivering the guest lecture. Now I welcome her for further guest lecture.
It's my immense pleasure to be here. I am so proud to be a Tamil Nadu Agriculture University student. I have learned so much of microbiology from this paternity. I am so lucky to have faculty like Tamil Nadu professors. I like Tamil Nadu stay and still I can speak Tamil. Wherever I'll go, everyone asks, are you a Tamilian and Telugu girl? I have so much of bondage to Madurai also, and all the faculty who worked during that time. Good morning to everyone, respected Professor N.O. Gopal sir, and respected Vizula madam, and all dear faculty and my dear friends. Today, my lecture on how the microbiomes we have to concentrate based on the microbiome, how we can modulate the crop genotypes. Actually, when we are talking about the sustainable agriculture is a buzzword nowadays. Everyone is taking care of the sustainable production. Uh, Previously, when we are talking about the microbial inoculants, in 1950s, we used to start utilizing only individual cultures, nitrogen fixers, phosphorus solubilizing bacteria, phosphorus solubilizing microorganisms. Now we are talking about the consortium of group of microorganisms, because now our challenge in agriculture production is nutrient use efficiency. If the nutrients are available in the soil 100%, the use efficiency is very less. That is the major challenge in agricultural production system. That's why now we invented that microbiomes can modulate the plant genome or plant genetics. How we can see one by one. If you see the population dynamics of globe, you can see the world population is increasing year by year. Then if you reach to the 2050, the population size may be 9.8 billions. But year by year, decade by decade, the cultivated area or net cropped area is decreasing. Then what are the factors which will influence if you use the net cropped area is less and the production should increase? But with the advancement of genotypes or with the advancement of molecular techniques, we could achieve the production, but the production is almost stagnated. You can see here, the year by year, the population is increasing, but our challenges is depleting the natural resources, land, water, and energy. Yield ceiling is irresponsive. If you apply 120% of chemical fertilizers also, we are unable to increase the yield level. Then one more challenge is sustained food security while elevating the hidden hunger. Almost 10 million people still they are unable to get the dinner, only they are satisfied by only one day meal. That's why still we have to search the innovative work, how we can increase the production level in normal net cropped area. That's why these are the technologies, innovations for future farming, molecular breeding. That's why I have taken this a topic, how the microbiomes can modulate the plant genotypes. Then crop biofortification, microbial technologies. All we as a microbiologist, we should accept. After Corona, everyone is talking about the microbe. Whoever don't know about the microorganisms, if you see the statistics of the production levels of bioinoculants in Indian level as well as global level, the drastic increase was observed in the statistics of the bioinoculants usage. In European countries, they have already started to produce in all the levels. They are using all the bioinoculants. In India, also drastically increasing. Asia Pacific also it is increasing. That's why it's a good sign to the microbiologist. We can exploit it, or you can extract the biodiversity, which will be having the good efficiency, which can work in all the climate changes. Then precision agriculture and enhancing the irrigation efficiency. Wherever the possibility management practices are there, there we can intervention with the microorganisms, and we can increase the population dynamics, and we can modulate the plant genotypes, and these genotypes can increase the production level. And then microbiome is not a new word. Already so much of work has been done with the human microbiome. We are considering microbiome with all the microorganisms like soil bacteria, fungi, and viruses that inhabit the particular environment, microhabitat. which will be having the positive interaction with the plants or host. It will be giving so much of resistance to the particular host. It may be human being or animal system, or it may be plant system. The vital for the well-being of the ecosystem or organisms, we can see the contribution from the microorganisms are very high. That's why every living system, the plant immune system will be increased by the microbial food. That's why we have invented now probiotics, prebiotics, and animal feeds. We are supplementing with the microorganisms. In everywhere we are using the microorganism, because the microorganisms are having so much of potential to increase the immunity level of the particular living system. Then what is microbiome? I told you can take any particular part of the plant or any particular system or organelle of the human being or plant or animal system. They have the particular biome which is related to the particular microhabitat, or which is related to the niche. Then these microbiomes requires a fusion of microbial diversity. Previously, when we are going to develop hybrids or different advanced genotypes, they used to consider only the genetical characteristics of the plant. But with the invention of the microbiomes, we can or we can take the consideration of the set of genes present in the rhizosphere. What now the student presented that rhizosphere is having the more microbial population when compared to the endophytic bacteria, because the rhizosphere is having more, it is a power of currency of root exudates. It always depends upon the photosynthesis capacity of the plant. If the plant biomass is high, if the photosynthesis capacity is high, the root exudation capacity of the plant roots are high. Because of that, the plant can recruit so many beneficial microorganisms because of the high efficiency of these root exudates. Root exudates are having all the organic compounds and inorganic compounds which can support the growth of the particular biome or microbiome present in the root system. Then these plants showing so much of potential microorganisms. If you study, so much of literature is available now. The student also has been presented that how the microbial dynamics are changing with the rhizosphere soil, or it can be managed with the management also, agronomical. How this microbiome can help. It will be helpful for nutrient uptake, as well as it can be protected from the pathogens and regulation of host immunity. The plant microbiome can influence the ecosystem functions. Then ecosystem services are intricately linked to plant functions traits, of which several are like mediated by the microbes, including soil formation, decomposition, organic matter decomposition, nutrient mineralization, and primary productivity. Means if the particular genotype is having the good beneficial communities in their rhizosphere, the plant is very fit to survive in any circumstances, whether it is having the biotic stress or abiotic stress. If the plant is having the good beneficial communities in their rhizosphere, the plant will be fitting in particular climate. That is the advantage with the microbiome. That's why now we are targeting the synthetic preparation of the consortium of microorganisms. We are introducing into the rhizosphere as a biofertilizers or bioinoculants. Then the plant can be competitive to grow in stressed conditions. Then core plant microbiome, why we are considering means it can, along with the plant genes, you can consider the microbial genes, because it is a reserve source of, because of the advancement of molecular techniques. Previously, when we are working in microbiology department as a graduates, MSc graduates and PhD graduates, we used to have only cultural techniques. So many microorganisms we are unable to cultivate in the plate. But now so much of advancement is there. Because of that, we can grow the unculturable microorganisms also. That's why we can take the consideration of this group of genes. These genes can help to modulate the plant. Here my simple concept to understand is, if you have any good friendship in the group, Okay, if anything happens to the particular person, he will take the help of our friends, and we will be sustaining, or we will be fighting against the particular situation. In plant modulation or plant genotype sustainability also, if you consider only plant genes, it may be difficult to sustain in this climate change conditions. But if you take the consideration of microbial genomes, the plant can adopt these genes or the help of microorganisms. It can sustain very well in particular condition. That is the main concept in modulation of microbiomes with regard to the genotypes. You can see here now the studies are available in literature. Every part of the plant, whether it may be the seed, or it may be the endospore, or it may be the guard cell, or those parts are having the specific biomes. These biomes, if you know about the potential organisms of the particular microbiome, seed microbiome, root microbiome, or leaf microbiome, we can consider these potential organisms, and we can explore those microorganisms, and we can make it as a consortium, and we can use for the external application of the inoculants as a consortium based. And nowadays the biofilms also fetching well in soil microbiology. Here in biofilms concept, what we are using, different types of combination of microorganisms like fungi, different types of bacteria. We are using the fungi as a base. We are coating all the microorganisms on the mycelial growth, and there we are making the biofilms. This will be sustaining in the soil. It may not be removed with the antibiotics, or weedicides or herbicides also can may not reduce the population of biofilms. That's why now everybody is talking about the consortium and biofilms. It will sustain very well. We can explore these microbiomes to prepare the synthetic bioinoculants as a group, and it will give so much of benefit to the sustainable production. Then when we are talking about the endospore or seed is the if the when we are talking about the resistance power, if we will get some resistance from our parents, like that only in plant also. Mostly when we are talking about the soil borne pathogens, the resistance against the soil borne pathogens or plant viruses, uh, the defense mechanisms will work well with the good seed variety. Or we will recommend anytime, if the farmer is suffering from the soil borne disease, first of all we will recommend the resistant variety. That's why if you target the genotype, or if you target the gene plant variety, if the plant variety is having the good population of the beneficial communities like the seed, from you can see the seed microbiome is giving the clear indication that if the particular variety is having the good beneficial microflora in the seed microbiome, it will be giving so much of resistance when it is going to start their life in the soil. That's why because of the good beneficial microorganisms, we can tell that 50% of soil burn seedling wilts can be reduced if the variety is having the good beneficial microorganisms, which can give the resistance towards the soil borne pathogens. You can see here I told if you explore the air microbiome or phyllosporic microbiome, endophytic microbiome, if you able to study the microorganisms which is present in the particular area, and you can explore the potential strains from the same genotype, and you can identify that among all the microorganisms which is potential, which is having the multi-trait effect. Multi-trait nothing but same microorganism. We all know that same microorganism may have the plant nutrient mobilization, then antibiotic production or rhitic enzyme production. If one organism is having all the traits, then it will give so much of resistance to the plant. You can see here the plant, if you select the right side plant when compared to the left side plant, if you identify all the microorganisms, the potential organisms, you can make as a synthetic biofertilizers or consortium microorganisms. If you apply the field, it will sustain in any climate, and it will give the good production when compared to only chemical fertilization. You can see here the model plant microbiome with emphasis on the below ground habitats. Now I told everyone focused on the above ground level only, how the plant can sustain, how they can manage the plant population, what are the management practices they can follow for sustaining in climate change. But below ground levels, only superficially we are knowing that. We don't know the deep level, how the microorganisms really giving the plant health, how it is influencing, what are the if you take consideration of the all the genome or genes present in the soil rhizosphere, if you consider all the things, then if you make it one place, then the sustainability of the plant will be increasing. Immediately you can reduce the usage of the chemical fertilizers as well as the fungicides. It is giving so many toxic levels in food as well as the soil. If you consider, at least as a microbiologist, if you recommend to the farmer, if the farmer is reducing at least 20 to 25% of chemical fertilization in commercial crops, we can reduce the soil pollution as well as leaching of the nutrients to the irrigation channels. That's why we need to focus how these modulations occur in plant genotypes, then how we can reduce the chemical fertilization as well as the chemical pesticide application in the soil. It can give the sustainable production of the plant. Then we all knew that how the plant microbiome mediated adaptive functions like direct mechanisms and indirect mechanisms the microorganisms will give. These are the nutrient mobilizations like nitrogen fixation, phosphorus, potassium, and zinc solubilization. If you consider the nutrient use efficiency, for phosphorus only the nutrients use efficiency is only 18%, 18 to 20%. Remaining 80% of phosphorus will be fixed in the soil. Along with the phosphorus, all the micronutrients also fixed in the soil. That's why if you use these microorganisms, it will improve the production of organic acids and it will chelate the fixed form of all the nutrients. It will mobilize to the plant, as well as it also increases the iron chelation as well as the sulfur oxidation. Then by application of these microbiomes, or if you increase the uh, uh, percentage of beneficial microbial communities in the soil, it will improve the biomolecule production like hormones, ACC deaminase, and different lytic enzymes, which will provide so much of resistance to the plant. Then nowadays, for example, now in Telangana state, we are facing in chili crop, there is much infection of the black thrips. Farmers are leaving acres of the land, they are unable to control the black thrips. But the last 2 years we are recommending like all the bioinoculants like Bavaria bassiana and Verticillium lacani, along with the biofertilizers application in commercial crops cultivation like chilies and maize. Then they are giving the feedback where they have used these bioinoculants or beneficial communities, they could tolerate, or the crop can tolerate the black thrips population when compared to only chemical fertilization. That's why nowadays farmers are ready to accept our beneficial communities or microbial fertilizers, but we have to develop the quality product. Whenever we are talking about the microbial communities, it is giving very good result under in vitro conditions. When we are recommending into the field level, it is not successing. That's why whenever we are targeting the beneficial communities, we should not do only microbial aspects. We have to combinedly take the consideration of all the crop production groups, as well as pathologists, as well as agronomists and genetics people. Then we have to do the interdisciplinary work. Then our microbial consortium or bioinoculants can work well in the field level. That's why whenever we are talking or any research project, if we are applying, they are asking about the interdisciplinary. Now this is the desperate need of research we need. We have to combinedly check that our microorganisms should fetch well with all the recommended practices which we are recommending for sustainable production. This is the now all the genomic tools are available to study all the microorganisms or microbiomes present in the soil, and we can consider previously actually my thought used to be if the population size of fungi is high in the soil, it may not be beneficial because the fungi population always it will be saprophytes. But we can consider the trichoderma. Trichoderma is a potential agent which can control the so many diseases and pests, and it is also working well, Pasolomyces or Trichoderma working well with the nematodes also. In particularly in my area in Telangana, all the nearby orchards of Telangana was infested by the nematodes. Their acres of orchards are removing with the infestation of nematodes. There, if you have the potential organisms which can control the nematode population as well as the soil-borne pathogens, really it is a noble work. That's why now we are concentrating these microbiomes where we can combinedly take the consideration of fungi, bacteria as well as actinomycetes, which can sustain very well in the soil as a group, not in individual group. That's why we are targeting the microbiomes now, and we are studying all the beneficial microorganisms, whether it is a protozoan or whether it is a fungi or bacteria or actinomycetes. We are combinedly taking the consideration of compatible studies, and we are applying as a consortium in different fields. Or this next. Here, how do plants harbor unique microbial or special microbial communities? How do plants shape special rhizosphere microbial community? Nothing, but they have. I have collected the literature. They have studied in Arabidopsis plant. They have studied the different genotypes having the different beneficial communities. Means now ma'am has asked about the genetics head. She was asked whether genotype can change the communities. Yes, really it will change because of the root exudation. I told different genotypes will release the different types of root exudates. Based on that only the beneficial communities arise in the soil. That's why they have reported that when they have studied the different genotypes of Arabidopsis, different species, they found that different genotypes promoted the different types of microorganisms. It's not same variety cannot having the same microflora based on their photosynthesis capacity or root exudates. It will change the microbial communities, and they observe that the root microbial communities of the plants are sufficiently depends upon the host genotype. It may not be common to all the genotypes. Next. The optimizing host selection of the root associated beneficial microbial communities holds a promise for the sustainable and increasing crop production. I told now the crop improvement group is able to give all the varieties, but they don't have any studies with relate to the microbial communities. That's why if they could give for each genotype what is the level of the communities present and beneficial communities present in the soil as well as the endophytic bacteria, then it will give so much of value to their work. And if you go in that line, it will give so much of evidence that how the modulation occur with the genotypes. Next. I told already because of these defense mechanisms present in the microorganisms, it will be utilized by the plant also. Like uh, whenever we are talking about the bacterial cell is having the nucleoid as well as the plasmid. If you target the nucleoid, the bacteria will survive with the plasmid. Like that only, if when we are going for cultivation of the crop, if the pests and diseases are destroying the life or plant, the plant can explore the genes present in from the microorganisms and it will sustain. That's why we need to focus in the plant microbiomes. It will give so much of information how the plant can survive because now our challenge is in sustainable production is climate change. That's why if you need to give the adaptive techniques for plant, then you can explore the microbial genes for genotype modulation, then it will help to sustain. Next. Next, I told all these reason is root exudates, because the root exudates can decide the microbial population presence or communities present in the soil. That's why root exudates are very important. Still, we are unable to understand how many types of root exudates are there, then how it will be recruiting the microbial communities based on their inference with regard to the nutrient management and everything, still in infant stage only. That's why if we concentrate in the root exudate proliferation in the soil, then it will give the clear indication that how the plant genotype can recruit the beneficial communities. Then next. Next. Then we all knew that always the positive interaction will be present for the soil, plant, as well as the interfaces. Now here, then how it will be negative as well as the positive feedback to the plant when we consider about the soil interfaces and microbial interfaces with the plant. Then negative interface, nothing but negative feedback will be given by the plant where the harmful or plant pathogens are present in the rhizosphere soil. That's why we are modulating the microbial population level with the inoculation of microorganisms as a bioinoculants like biofertilizers and biopesticides. There you can inhibit the harmful pathogens in the soil and you can increase the beneficial communities in the soil. It will directly give the positive feedback to the plant and the plant can sustain in the soil. Next. Next. Then holobiont as heritable unit of the selection, because holobiont is the assemblage of the individual and as well as symbiotic relationship with the different microorganisms. I told already, if the plant is having the plant microbe interaction is positive, the microorganisms will give the nitrogen, phosphorus, or defense mechanisms and every support to the plant. The plant in turn will give carbohydrates to the microorganism. That's why if the symbiotic relationship of the plant and microbe is best, then the plant can sustain in the soil. That's why we have to explore these benefits from the microorganisms. Next. Next. Here, I told if the rhizosphere soil is having the keystone species. Keystone species nothing but what we could see now with Robbins experiments. Population densities are high, but when we are talking about the biochemical characterization and molecular characterization, only two organisms are dominated, like Enterococcus as well as Bacillus he has given. Like that, if we identify the alpha proteobacteria or proteobacteria, which communities are benefiting more, if you completely replace the rhizosphere soil with these beneficial microorganisms, then it will give so much of fitness. That's why the plant is very clever. How means when the unavoidable situations or the climate change occurs in the soil based on the physical characteristics and chemical characteristics, immediately the plant will recruit the beneficial microorganisms. Through that only it can sustain. That's why definitely we should ask the farmer, he has to use the microorganisms. Okay, how we have changed our food habitats after corona, means everyone started taking the protein food. Previously everyone is taking okay, normally carbohydrates like that, but no, now if you consider the statistics of the disease pattern of the corona also, whoever having the high resistance power, they have tolerated the corona. And during the second season, so many people were died, but who are having the resistance, they only sustained. Like that only in plant also, if you are giving the resistance through the microbial inoculants or through the application of organic amendments, soil is having so much of organic carbon, then because in southern states or tropical countries, the organic carbon status is only less than 0.5 percentage. If you are applying the bioinoculants also, there is no sustainability of the bioinoculants in the soil. That's why always, if you go for the integrated nutrient management along with the organic and chemical fertilizers, it will promote the plant beneficial microorganisms and plant beneficial communities, and it will give more sustainability. And the plant usually gives so many networks of the beneficial microorganisms, and it will give the benefit to the plant. Next. 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 Okay, next. Actually, here now we are talking more about the ecosystem services. Ecosystem services, nothing but we all knew that when we started the green revolution, we should convince to the farmer whether he is not agreed to apply the chemical fertilizers. But now we are talking about the second green revolution. Second green revolution, nothing but here, whatever the produce we are producing from the farm or from the field, agriculture field, it should be used in the soil itself, or it should be used in the particular field. That's why here our concentration is the natural resource management. Where when we are talking about the natural resource management, what we are going to create the artificial ecosystem. Artificial ecosystem, nothing but we are not going to prepare from the chemical point of view. I am talking here, if you know about the plant microbiomes or which genotype is having the which microbiome, you can potentially explore the microorganisms, and we can make as a consortium of microorganisms, and we can replace the rhizosphere soil with these artificial or synthetic microbial populations. Then the plant can sustain very well, and it can fit in any climatic conditions. But its success in open system is challenging. What I could tell, in when we are working with the in vitro conditions, it is working well. But when we are treating in the field, the microbial populations are not sustaining. That's why we have to concentrate more to work with the field conditions, with all the climatic change or pH, salinity or sodicity, whatever it may be. That's why if you consider the field conditions, then our microbial communities may replace the normal conventional methods of the cultivation. It will give so much of benefits to the plant. Next. These are the schematic depiction how we can explore the microorganism from the different microbiomes, how we can use these synthetic microbiomes, we can synthetic bioinoculants we can produce. Everybody knows as a microbiologist here how we are exploring the microorganisms. um, it is the mandate now in agriculture. Definitely we should go for the synthetic bioinoculants in all the fields and all the crops cultivation. Then only we can restore, and after 10 years we can give the good field conditions to the next generation. Next. 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 Okay, here I have given the picture that different factors affecting the plant microbe interactions. Whatever the synthetic microbiomes I am telling, if you apply in the field, the microorganism should give the resistance to the plant through the root exudates. The plant can sustain under abiotic stress like salinity, cold temperature, drought, and heavy metal concentration, and other biotic stresses. You can see how the different microorganism causing the different diseases. If your plant is fit with the beneficial communities, it can reduce the stresses of biotic as well as abiotic stresses. And these biotic stresses, mainly the farmer may not have any control measures against the viruses, nematodes, as well as fungi. That's why if you use these microbiomes to produce the different genotypes, then it will give so much of fitness to the plant to sustain in these biotic and abiotic stress conditions. Next. Here, a model for the plant microbiome response to microbial inoculants. I told if you inoculate your plant, okay, with the plant microbiome, the immediate shift can occur in the soil. If your seed is having the more beneficial microorganism, you can expect in short time the shift of beneficial communities in the soil. Because of that, you can restore all the good conditions in the soil, and the soil productivity will be increased. Next, these are the some of the literature I have collected from the how these microbiomes assisted breeding to the understanding cultivars or genotypes depending upon assembly in cucurbit and pumpkin plants. The next, in this study, next they have studied how genotypes, how different pumpkin genotypes influence the beneficial communities. They have given that clear indication that each genotype is having the own microbiome. It may not be common in the same genotype. They have studied uh, different genotypes in pumpkin, and they quoted that enterobacteria is 40 to 83% in particular organisms. In wild type, more communities are there when compared to the normal varieties. Next. Then Elizabeth et al. 2020, they have studied about the tomato genotypes, and these tomato genotypes also modulated, and they have given that genotype accounted for 10% of variation in root microbiota. Two bacterial families like Bacillacea and Rhizobiaceae were significantly enriched in the root endosphere, at least 6 of the 8 tomato genotypes. Here, we could understand that the genotype can modulate the beneficial communities, and it is every genotype is having the own microbial communities when compared to all the genotypes. That's why whenever we are targeting the microbial populations or microbial communities where there is no much disturbances, like forest lands or undisturbed rhizospheres may have the good beneficial microorganisms when compared to the cultivated lands. Next. Here I am concluding my topic. When we are talking about the genotypic modulation with regard to the plant microbiomes, we can explore these microbial genes or genomes present in the rhizosphere soil. When compared to the endophytic bacteria, rhizosphere soils are having more microbial densities. That's why we can explore these microbial densities or plant microbiomes to improve the crop improvement with these genotypes. It will give so much of inference, or it will give so much of evidence that our genotypes can sustain in present challenge of climate change. It will be helpful to give the sustainable production, and it will increase the nutrient security of the food. Thank you. The topic is open for discussion. If you have any doubts, I can try to.
Thank you, madam, for a very elaborative and also thought-provoking guest lecture. I think many students will be benefited and the staff members will also benefited. As you said, the watermelon and tomato, the response of the microbes, I still remember there are some synthetic bioinoculants for flower crops.
Yes.
Especially for increasing the color of the flowers like petunias and
Yes,
other
sir.
thing. Whenever they market the seed, they also market the mixture.
Yes.
They say it is a nutrient mixture. I saw in the US. And when you put in pot culture, you put that mixture, then the color of the flowers will get increased. So there is a very long way. Synthetic bioinoculants are helpful. Similarly, a lot of studies have been already done on legumes.
Yes, sir.
Genotypes
Flowers.
responsible to the microbes, not only to Rhizobium, many other organisms.
Yes.
So it is a quite interesting and important concept where students to take up for the research and also the proposed projects and other things also. I appreciate your weddings like nutrient currency also.
Yes.
That is, that was somewhat triggering me. Nutrient currency is a very good word. You used it for your presentation and other thing. And how best we can modulate these microbiome to the extent in rhizosphere or phyllosphere or in both? Any other
Yes.
suggestions, madam?
Actually, sir, if you target the rhizosphere, it will be better, sir. If we target the phyllosporic bacteria, the sustainability or the presence of the microbial population, if you apply also, the sustainability is big challenge, sir. If you target the rhizosphere microflora with the beneficial communities, then it will give the better results when compared to the phyllosporic bacteria.
Thank you, madam. Any other question? Now we open for discussion. Students should ask questions. First, from the madam student, Lavanya.
Uh, Good morning, ma'am. Thank you for delivering a good lecture. Actually, I'm also working in the same area. So, different genotypes will accumulate specific microbial communities. So, how we can study those
Act-
techniques?
Rice genotype. Okay. Actually, now we have the different planting systems and we are targeting, for example, in case of the rice. Okay, fine varieties are there, bold varieties are there, but mostly we are recommending the fine varieties. But the fine varieties are having the more pathogen and pest sustainable susceptibility. That's why when we are targeting the microbial communities, you have to take consideration of ruling varieties in particular area. Then what is the acreage present in particular variety, and you can target those rhizospheres. And first you have to explore the microbial communities actually sustaining in that particular field. Then you have to comparatively study the all the characteristics or all the traits of these microorganisms, and you can consider the only potential isolates among all the groups, and you can make as a synthetic microbiomes. Synthetic microbiomes nothing but you are going to give the explanation of each genotype how it is modulating, whether bold varieties or fine varieties, and you can use all these microorganisms in different rhizosphere soils, and you can make it as a one formulation and you can study. That will be the better option as my knowledge.
Thank you, ma'am.
Good afternoon, ma'am. Thank you for the lecture, ma'am. Most of us here are working on endophytic microbiomes, ma'am. So we have different endophytes like leaf associated or nodule associated. Uh, During my ORW presentation, I had one question for me, ma'am. How do we designate them as the particular region associated microbiome? Because these endophytes are localized on every parts of the plant.
Yes.
And the particular microbiome that the work that I am doing, the particular endophyte was found in the rhizosphere, was found in the seed, was found in the bulk soil, and also in the nodule of the plant, ma'am. So how do I designate which part of the plant it belongs to? Because it belongs to different microbiomes, but how do I specifically designate where it has a higher or bigger role to play?
Actually, you see, whenever we are talking about the endophytic bacteria, so many microorganisms, if the microorganisms colonized the root, it will translocate to the leaf. Definitely. For example, if you consider the Pseudomonas, it will translocate. Maybe it will be present in the soil. Mostly soil is having Bacillus, Arthrobacter, and Pseudomonas. These three organisms we can find in all the microbiomes. When we are pointing out of the particular microbiome, how it will sustaining in particular area. Okay, but when we are talking about the environmental pollution, for example, when we are talking about the soil microbiome, okay, Soil microbiome is very large. It will be having so many microorganisms or so many populations also will be there. If you consider endophytic, what you have discussed, pillosphere or inside the seed or inside the plant, there is much competition may not be there. Did you get my point? If you are talking about the soil rhizosphere, there will be so much of competition with other organisms as well as plant pathogens. You cannot find all the plant pathogens in the seed as well as leaf. There is no much competition. All we know as a microbiologist, if any adverse climate is prevailing, then the microorganism will start triggering their traits. Unnecessarily, the microorganism may not release the antibiotics or endotoxins. No, if we all knew that if you are giving 100% of nitrogen application in the soil, the rhizobium may not form the nodules. No, because it may not waste their ATP, 16 ATP, for synthesizing one molecule of nitrogen to 2 molecules of ammonia. Like that, the microbiome which is present in the soil will be very large. That's why when compared to the endophytes. That's why if you consider only soil microbiome, you can explore more type of microorganisms which will be competing with other plant pathogens as well as other microorganisms. That's why if you target other endophytes, it may not sustain also in soil. Okay, if you told no, because so many people are also working with the endophytic organisms. These endophytic organisms, if you select and if you prepare as a synthetic microbiomes, sometimes it may fail in the soil. That's why if you target the soil microbiome in different genotypes, I told different genotypes will release different concentration of root exudates. It will recruit different microorganisms. That's why, in my opinion, if you target the soil microbiome, it will give best result when compared to the endophytic bacteria.
Thank you.
Thank you for your wonderful lecture. Uh, Actually, the thing is, in case of the food security, so many microbes like PGPRs are from the category like Enterobacteriaceae family. As you have given in one of your research paper, in the Cucurbitaceae family, around 40 to 83% were from Enterobacteriaceae family.
Yes.
But in the case of aspect of food safety,
Yes.
Enterobacteriaceae is
The
like
foodborne
very
disease, it causes
many opportunity as well as many foodborne pathogens are there. So considering the foodborne food safety aspect, if you are giving these as bioinoculants, what it becomes some opportunistic way for the transfer of foodborne pathogens or like that?
Actually, if you see the literature, everyone, everywhere, any microbiome is having more Enterobacter populations are coming. He also mentioned about Enterobacter. We also deposited 2 or 3 Enterobacter species newly isolated from different fields of harsh climates of India. Here, first of all, really it is an opportunistic pathogen. Okay, Pseudomonas syringae also Aerogenes also opportunistic pathogen. But when we are talking about the resistance, okay, we are talking about the plant fitness here. Okay, we are not talking about the product which will be coming from the rice. We have to consider that when we are applying as a biofertilizers, we are applying in the soil. Directly, we are not keeping with the food like a grain. Once if you have isolated the Enterobacter, it is an opportunistic pathogen. First of all, you have to confirm that whether it is causing any pathogenetic efficacy on the plant. Then again, you have to check for the grain or any food economic part, whether it is sustaining in final product or not. That one should confirm before recommending as a commercial product. that will be.
Grain crops,
Mm.
the crops like cucurbits and some fresh fruits
Yes.
and vegetables are there. There it's high possibility of transferring the foodborne pathogens if we are going for inoculant technology.
That's why what I'm telling. No, if you apply as a synthetic microbiomes, synthetic inoculants in the soil, we have to confirm that whether it is sustaining in pumpkin or not. That studies I couldn't find. Future we can try like that. We can apply this Enterobacter in the soil, and we can study the populations present in the.
Why there is always some link between clinical pathogens, like so many are there, not only Enterobacter, Pseudomonas, and so many are there. Those are very good PGPRs as well as they are opportunistic pathogens. So why it is like that in so many stressed soils?
Because
Like
yes,
stress.
because no, you consider the microbial populations, how it will be. These are so many opportunistic pathogens. Why means we all knew that you see Helicoverpa will cause the pink boll worms. We have developed the BT gene. Okay, then now all the BT genes are failed. Bolgod 1 failed, Bolgod 2 failed, because no, the microorganisms continuously they develop the resistance mechanism. Because of that, if you are able to identify the resistant genes which are helping to provide the resistance, then future we may think we can identify the particular genes and we can silence that genes. It may not express and it may not cause the diseases. Whatever you have expressed, it is a good question. When we are talking about this Bacillus and Pseudomonas, we are telling it is a beneficial to us, but those are potential human pathogens also. But
Species.
yes, potential human pathogens. But still, we are exploring because we are concerned about our soil. That's why when we are talking about the soil beneficial microorganisms, it should be the chain, a link of chain. You have to confirm whether it is having any harmful effects on the human being or animal and everything. It is related.
Thank you.
Thank you. A good discussion.
Thank you.
I want to add only one point for her query. As far as the watermelons and other things are concerned, a lot of work has been done to reduce Enterobacteria by various. And you can visit UC Davis Food Safety site, where in the fresh cuts and also the whole fruits, they have done enormous work to reduce the enterobacterial infections in the food safety point of view. It is a very good question, valid question also. But as far as the fruits are concerned, either fresh or fresh cuts, whatever may be, they should be processed in a proper manner to reduce these contaminants and other thing. Any other questions? So if there are no other question, I thank Madam for their excellent presentation. On behalf of the department, I request Dr. M. Jnanachitra to present a memento to Dr. Triveni Madam. Thank you, Madam. Now I request Jnanachitra to propose a formal vote of thanks.
Good afternoon, everyone. Respected Professor, Madam, and dear students. On behalf of our Professor and Head and other professors, I thank Madam for coming over here to conduct Viva Voce and to deliver this guest lecture. Thank you. Thank you so much.